Hello students, thank you very much for watching my lecture, Selected Applications of Polypeptide Structures. In the previous lectures we have seen how amino acids can be connected together to peptides using solid phase synthesis. We have seen how the synthesized peptides can be connected together to form larger polypeptides or even whole proteins. And finally, I've very briefly discussed how recombinant DNA technology works, with which you can use a bacterial expression system to fabricate your protein of interest. What I would like today in this lecture is to show some selected examples where the different methods of peptide synthesis and protein fabrication have been used to highlight some particular examples. The first one I would like to discuss with you are coil motifs. Here you see small peptides that have been chemically synthesized by solid phase synthesis. These peptides, they form alpha helices, and these alpha helices, they assemble together in this coil motif that you see in the upper panel. And when you look at this panel, you see that one helix turns around the other helix. That's the so-called coiled coil motif. And this coil motifs, they aggregate together into larger fibers. So several of these coiled coils come together and they cluster into these fibers that you see here in the graph. And these fiber networks, for example, have been used to culture cells inside. This is purely synthetic materials, and that's why these materials are very pure and for that reason also interesting to be used as three-dimensional cell culture medium. Another example I would like to show you is from my own group. This is not yet published, but at the moment at least I find it exciting, and what we have done here is we have made a nanoparticle system, and this nanoparticle system can be activated by ultrasound, and the idea or the purpose here was to activate antibiotics by ultrasound using such a gold nanoparticle format. We have synthesized large gold nanoparticles that contain a peptide sequence that contains non-natural, or at least not the standard amino acid. This motif, that's the green one, contains two D amino acids, D-alanine, and D-alanine is also present in the cell wall of bacteria, and this D-alanine motif, that is recognized by the red peptide shown here. This is a commercial antibiotic. It's called vancomycin, and it kills the bacteria by interfering with the cell wall synthesis. In this case, we used the large nanoparticles with the green peptide, and we made the small nanoparticles with the drug on the surface. The big particles were used to inactivate the small particles carrying the vancomycin, and once they form this complex, the complex is inactive, and by ultrasound the two particles are segregated from each other, and then the small particles get active. Let's see how the system looked like in reality. So we have synthesized the two particles. Again, as mentioned before, the synthetic peptides and the natural peptide drug, play very important roles. Here you see the big and the small particles. You see that the peptide motif on the big particles is able to assemble the small particles around it. Once you let the solution stand for longer, then you form particle networks that are induced by the recognition of the antibiotic with the peptide sequence. And once you apply ultrasound, the aggregates get disaggregated, and then the small uh, gold nanoparticles with vancomycin on the surface bind, for example, to Staphylococcus aureus, and by this, as you can see on the right-hand side, kill the bacteria. On this slide, you again see some technical details. You, for example, see how the nanoparticles separate upon ultrasound, and then a color change is induced that shows that particle separation takes place. You also see bacterial growth curves that show that the nanoparticle networks can be activated once you apply ultrasound to the system. You also see that the ultrasound itself doesn't kill Staphylococcus aureus, so Staph aureus is quite resistant to the ultrasound conditions that we apply here. And on the scheme below, you see how the different systems can kill bacteria or don't. So there are also the respective controls shown, and the final right plate shows that indeed the ultrasound plus the gold nanoparticle assembly uh, turn on the function of the antibiotic, and most of the bacteria are killed. In this case, now I've chosen another example. In this case, this is a small chemically synthesized polypeptide system that is able to regenerate or to induce nerve growth. This is especially important when it comes to regenerating nerve tissue. For example, uh, if the spinal cord has been injured by a trauma, uh, here gels have been developed that should induce new nerve growth, and by this bridge the gap that had been implemented by the trauma, and regenerate nerve growth and thereby also nerve function. What has been done here is amphiphile has been synthesized by using solid phase peptide synthesis. When you start watching at the structure, you see that there's a long alkyl chain attached to one end of the peptides. Then there is a glycine alanine motif shown here in red. This glycine alanine motif is meant to form a beta sheet structure, so to make a strong assembly in aqueous solution. And on the top of your system, you see the IKVAV motif. It is a motif found in laminin, and this motif can direct the differentiation of NPC cells into nerve cells. And with this multiplied motifs on this rod-like micelles that are adopted by the peptide amphiphile, these type of structures are very soft gels. They can be injected into a traumatized nerve tissue, and by this it is able to regenerate the nerve growth, as can be shown on the next slide by this animal model. So here you see a mouse that had been, the spinal cord has been cut, and then the chain integrated. Both of showing that nerve connection and uh,